There's a weird part of your spec that asks you to talk about post-translational regulation. So we've looked at pre-translational regulation in terms of splicing, and we've looked at transcription factors in terms of pre-transcriptional regulation. But post-translational regulation you've learnt about the whole time that you've been in biology A level. Every time you see a protein having its sort of structure or its patterning or its ability to do something change, you are regulating that protein post-translationally. It's been translated, you've got your protein, you're regulating its activity. There's a few examples they want you to know, and that's what I'm going to do here, is that I'm going to put it in context of something you've learned about, make you realize that you knew this all along. So as the example I'm going to use is the fact that adrenaline can cause glycogenolysis. You'll all be happy with the idea that if there's loads of adrenaline around, you'll need lots of glucose and lots of ATP production. So, therefore, let's look how it works. You should know that adrenaline can bind to its receptor, which is a GPCR, a G protein coupled receptor. And by binding to the adrenaline receptor, your G protein gets released. Your G protein gets released, and it can sort of bob along to this enzyme that is sort of hitched into the membrane, which is adenylate cyclase. So you should definitely know about adenylate cyclase. Sometimes we don't include the G protein, but I've given you all of it. So this receptor being activated by its ligand or the adrenaline is post-transcriptional regulation. It usually holds onto this thing, but when something's bound to it, it no longer holds onto it. That then can go and interact with our adenylate cyclase enzyme, which originally isn't active and then gets activated, and that therefore is post-transcriptional regulation again. We have regulated that after post-translational regulation, we have regulated after translation has occurred the activity of adenylate cyclase. You then learn about these second messengers being made, of which the example you always learn is CAMP, which is cyclic adenosine monophosphate. And how does CAMP work? CAMP, shockingly, goes and post-translationally regulates another enzyme called protein kinase A. So what ends up happening is CAMP binds in this little area here, this little pocket, and that activates it. And you have protein kinase A's activity increasing. But what's really cool about this thing is this enzyme has other enzymes as its substrate. So this enzyme, with the use of ATP, can actually stick phosphate groups onto other enzymes. And what those phosphate groups do, because they're quite highly charged, is they can actually cause conformational changes in other enzymes. So this, say for example, this is your glycogen phosphatase, which is what breaks down glycogen. So this is our glycogen phosphatase, which breaks down our glycogen. In this shape, glycogen no longer fits into its active site, so it's not active. After the activation of the adrenaline receptor, we get CAMP production, pKa activation. pKa shoves a phosphate group onto this enzyme. That changes its active site so that it now fits glycogen, and we can break down glycogen into glucose. Through a whole step after step after step of post-translational regulation, we have got to our end goal. We've got to our end goal of one thing causing another thing, lots of different proteins and lots of different processes regulating each other. You've learnt about post-translational regulation the whole time through A level. I'm just showing you that you can do it and introducing you to the idea of kinase enzymes, which can stick phosphate groups onto other enzymes and either activate or actually inactivate them. If you get the premise, I think that's all you're ever going to need.